which exists between you and us. Okay, audience, hope you all of you have joined. And today we have one very eminent, one of the young promising scientists of this country, Professor Subi Jacob George, to offer a talk. And unfortunately, he hasn't given a title of the talk. I don't know why. So I cannot announce the title of the talk, which anyway, he will be announcing soon enough, I suppose. Okay, Subi actually is our neighbor. He is from Piravam. And one more thing that I would like to say is, which is not evident from his biodata, is that in principle, Subi is our alumnus because it is Cochin University of Science and Technology which awarded him the PhD degree. Though he did his research at National Institute of Interdisciplinary Science and Technology at Trivandrum, his registration was with CUSAT, and I was, deeply involved with his career at that time. Okay, Subi has his major interest in organic and supramolecular synthesis, bio-inspired fuel-driven living and transient supramolecular polymerization, organic optoelectronics, etc. And one thing that I don't see here is his deep interest in self-preparing polymers. And to talk about his academic background, he got his MSc from Mahatma Gandhi University. And during the final semester, he joined for project at NIST, where he got really interested in research. Actually, he once told me that he was actually aspiring to be an engineer once upon a time, but once he started learning chemistry, he understood that chemistry has more exciting opportunities to provide him rather than an engineering job. And that's how he came to chemistry. Anyway, his short period at the project student at NIST Trivandrum was really helpful, and he decided to join the PhD program there under the, able mentorship of very famous scientist, Dr. Ajay Ghosh. And fortunately for us, his registration was with Cochin University of Science and Technology. He completed his PhD early, and then he left for his postdoctoral work in Netherlands. And he actually worked with two of the very famous scientists there, Major and Schenning. And after that, 2 or 3 years of postdoctoral work there, he decided to return to India, which was really good for India, I suppose, and joined JNCASR in 2008 as a faculty fellow. And he was promoted as a full professor in 2010, 2008, I guess. And there are so many, accolades that has been showered upon him. A few of these include Shanti Swarup Bhatnagar Award for the year 2020. So he's one of the youngest Bhatnagar Award winners now, because the second award, 2021 award has not been announced yet. He's also the recipient of Swarna Jayanti Fellowship in 2017. He's in the editorial advisory board of Organic Materials, the theme journal, which of course is a very famous one. And he's also the associate editor of Chemical Science and RSC Journal. Okay, with this brief introduction, I invite Professor Subi Jacob George to present his talk for the benefit of all the participants. Subi,
yeah,
it's over to you.
Yeah, thank you. Let me start by sharing the slide. Can you see it, sir? Can you see the slides?
You can see the slide.
Yeah.
We can see you too. It's really nice.
Yeah. Thank you. So good morning, everyone, dear teachers and students. Good morning to all of you. So at the outset, let me thank first Dr. Sebastian and Dr. Srimaran sir for inviting me to this MATCON 2021 conference. So I'm very honored again to get introduced by Prathapan sir. I got this honor once 2 months back in an international conference organized at Cochin. Thank you for very kind introduction, sir. I'm as Prathapan sir mentioned, so CUSAT is always close to my heart because I had the opportunity to get my PhD registration done at CUSAT, which was very crucial for me because my PhD work went very well, and that gave an opportunity that I could finish within 4 years, and then move to a postdoc. That was very important for my subsequent growth. So that way, CUSAT, it was very nice to get registered at Cochin University of Science and Technology at that point of time. And also before that itself, once I finished my 1997, when I finished my undergraduation. So I wanted to do a kind of research-oriented education, a master's. So 2 departments, which is close to Piravam, was obviously MG University campus and CUSAT campus. So I applied to both places, and in fact, I got admissions in the chemistry department of CUSAT. But then, yeah, finally, I decided to join School of Chemical Sciences. So at that time also, so I got many opportunity to visit CUSAT chemistry department many times during my student days. So many familiar faces are there. As I mentioned, Prathapan sir was my mentor and co-guide, also the examiner for my PhD program. So thank you. I'm like, he has given a lot of inputs. I still remember some of the questions he had asked during my PhD defense at the CUSAT campus. And also many friends in Srimaran sir, and then also my seniors like Dr. Manoj, also my contemporaries like Dr. Sindhumatai. So many faces are there in the chemistry department. So very happy to be back, happy to give a talk in a conference organized at the chemistry department. So with this just brief introduction, let me go to my topic. I'm sorry that the title was missing from the program. So, but as you know, I will be talking about kind of materials which we have been working in our lab for probably last few years. This is called supramolecular polymers. So we have a basically a supramolecular chemistry group at Bangalore, where we work at the interfaces of like organic synthesis and molecular self-assembly with the ultimate aim of making functional materials. And most of the time, one of the component in our materials is a kind of organic biconjugated system, so that we get kind of optoelectronic functionality to the materials we make. So the title of the conference is obviously Materials for the Millennium. I know that the kind of materials I'll be talking about is not as popular as graphene, but then it's a really emerging materials. Probably this field is active only for last 2 decades, but within these 2 decades, this kind of materials has shown a lot of promises. Okay, so my objective is to introduce to you this field. So I mean, I have given talks in recent times about this topic, but excuse me, but I'll be giving a kind of overview on this kind of material, that which is called dynamic supramolecular polymers. And I know that in CUSAT, a lot of activity goes on polymer chemistry. While we study out, while we work on these materials, there are a lot of similarities with polymer chemistry or polymers. In terms of its design principles, in terms of its, when it comes to address some of its challenges, most often we have to go back to the polymer chemistry books and learn from there and apply that into this kind of materials. So the materials we are going to talk about is supramolecular polymers, which I call it as it's an important step towards making adaptive materials or biomimetic materials. So I still feel in this century, we will see very interesting applications of these materials. We'll wait and see whether it will be called as the materials for the millennium. But then it has a lot of promises. So I show one slide here, one graphics here, which is kind of animation to give you a feel of these materials. So as you can see that this also contains long, so when these small molecules, it's coming together by weak interactions, and they form long polymer chains, okay? And long chains, which is very similar to classical polymer chains. Only difference is that in classical polymers, the monomers are linked together by covalent bonds, as we know that. But then in this case, they are linked by very weak interactions. Very weak non-covalent interactions. So sometimes if you want to make these materials, we need directional interactions like hydrogen bonding, or I mean, it can be guided by some additional directionality into the system. So I'll be talking about these materials, what are the challenges at the early stages of these materials, and where this field is heading. So the whole purpose is to introduce to this class of materials and further opportunities and challenges in this particular field. And I'm sure all of you know that last year we celebrated the 100 years of micromolecular chemistry. And within 100 years, this polymer chemistry has evolved to such an extent. We cannot think about a life, we can think about a society without polymers. That much impact this science had when Hermann Staudinger proposed in 1920 that it is a micromolecular. What is micromolecular chemistry? It's all about repeating units, monomers coming together and forming macromolecules. So I think it's very appropriate. Even like 100 years later, we are talking about something different, which is in fact a branch of polymer chemistry, I would say. So why this field has attracted a lot of attention in the last 2 decades? So as I mentioned, these are formed by weak interactions. So once it is by weak interactions, one of the advantages that we can make them completely reversible, because weak bonds can be easily broken. So we can probably it's an opportunity to recycle the complete polymer back to monomers by breaking the bonds which are connecting between the monomers. Okay, that's one opportunity. And also this gives, just like if you look into nature, I will come to some of the examples. If you look into the biological world in our cells, most of the assemblies are formed by the weak interactions, or most of the functions, or performing most of the functions. This kind of assemblies, which is similar to the polymeric assemblies, which is similar to the polymeric structures, are made up of weak interactions. So as a result, one can imagine that we get an additional property of dynamics into the system. So what I mean by dynamics is, if you look into one polymer chain here, these monomers can go out of the polymer and come back to this polymer chain. So that is called monomer exchange dynamics. So the dynamics, the rate of exchange depends on the molecule design, the structure of the molecules, which can be tuned from seconds to hours to microseconds for a synthetic chemist is possible. So this dynamics gives an important property to most of the biological assemblies, that is self-repairing or self-healing. If there is a damage, this dynamics will help the material to self-heal. So if you can make synthetic materials similar to this biological assemblies, one can imagine probably we have a adaptive materials, self-repairing material where the damages can be self-repaired. So you can imagine there are probably compared to the conventional materials, it will have an advantages of recyclability, dynamicity. That means it will come with a kind of self-healing or self-repairing properties. So these are the possibilities one can imagine. So even nowadays, a lot of attention has been made to recyclable plastics. That's very important for a sustainable society. And in terms of recyclable plastic, there are many researches going on. But one of the approach people are thinking, even can we use supramolecular polymers as a kind of recyclable plastics, where can recycle the monomers and reprocess it again? A concept quite different from a kind of biodegradable concept. So the basic of supramolecular polymers is obviously supramolecular chemistry, which all of you are aware of it. So the chemistry was defined as a chemistry beyond the molecule. And for people to start, the pioneers in this field, they got always inspired by the nature or the cellular world to make this chemistry, to start this supramolecular chemistry. So the chemistry is chemistry beyond the molecule. It's more than one molecule. It's an assembly of molecules. So if you, as I mentioned, inspiration is from cell. If you look into the cell, like a DNA or lipid bilays or protein assemblies, most of these assemblies in nature, in cell, are made up of these weak interactions. Either it can be a van der Waals interactions, hydrogen bonding interaction, pi-pi stacking interactions, or anything. And another aspect, if you look into the cell, is it's about dynamic nature of this material. This is a YouTube video which is made by a scientist in Harvard University. If you look into the YouTube inner life of a cell, which is made to convince you how complex is our cellular world. And these are the, again, a graphic, kind of animation of a microtubule, a protein which is very important for the cellular motion and so on. So you see that these microtubules are formed from tubulin protein monomers held together by weak interactions again. And you see that they continuously assemble and disassemble. So it's like a polymeric chain which is forming and disassembling by kind of external reactions. So that assembly disassembly process is very important to perform the functions. So you can see that if you look into biology, it's not a surprise that chemists at some point of time want to mimic some of these aspects in synthetic materials. And again, all of you know this field, supramolecular chemistry, started sometime in 1970s. And these three gentlemen got Nobel Prize in 1987, Cram, Len, Pedersen. So, but they were working on very discrete complexes. So their objective was to mimic something like protein substrate interactions in nature. So they were working on 1:1 complexes where there is a host which can bind to a guest molecules. So if you look into it, they are very discrete system. They will, if you take a mass spec of it, you will get one peak in the mass spec. So this is, so well-known examples are crown ether metal complexes, carcerans, cavitans, all of you study in supramolecular chemistry basics. So, so the principles behind this beginning of supramolecular chemistry was molecular recognition. So we know that the right shape of the guest or right shape and size matters. Like crown ethers, they have high selectivity. So the principles were molecular recognition, and they wanted to maximize the association constant. So this is a slide I have taken from my PhD thesis, which I submitted in 2004. So you can see that the energy involved in all these weak interactions are like a kind of very small. You can look into the ion dipole, hydrogen bond, everything. But then beauty is that although they are very weak, which helps us to make them reversible, when they come together, a kind of, in scientific terms, we can call them something called cooperativity. And I always show this picture where Gulliver constrained by a multitude of weak bonds. So even though they are very weak, when they act together, they are very strong. They can stabilize structures like DNA, protein assemblies, like in biological structures. So although they are weak, when they act together, they are very strong. So that was the basics of supramolecular chemistry. And because of this cooperativity nature, people immediately realized that the supramolecular chemistry, especially for organic chemists, is a powerful tool, or it's a powerful bottom-up approach to make materials. So you can synthesize molecules and engineer them in such a way that with weak interactions, they can assemble, they can form extended assemblies, and they can form materials. This was one of the earlier Rosette assemblies by Weisseitz and co-workers. And so that was a realization. Probably that is a realization happened probably the beginning of, first 20 years of supramolecular chemistry research. And with this realization, people have tried to make functional materials, especially organic materials, which made up of supramolecular chemistry. And the kind of material, supramolecular polymers, which I'm talking about, it's basically originated from this approach. This idea of making nanomaterials using a kind of bottom-up approach. So the supramolecular chemistry has evolved, as I mentioned, to even if you look into the host case complexes, in 20 years of time, you can say that we can even make molecular flowers by a kind of programmed self-assembly. So nowadays, just like organic synthesis, people started calling it as non-covalent synthesis. You can do retrosynthetic analysis here, and you can make even complex or bigger containers where we can do catalysis, which can be used to stabilize the intermediates and so on. So you can see that with just as we grow, like 20 years, the complexity of the supramolecular designs have increased. But I'm not going to talk about this particular part of host case complexes. As all of you know, we'll move to the materials. And I just want one more example, which is reported from Tokyo. It's sometime in 2006, to convince you that this is a powerful method. Like supramolecular chemistry is a kind of wonderful bottom-up approach to make nanomaterials. This is a kind of hexabenzopyronene, which is a well-known P-type organic semiconductor. You make it a kind of amphiphilic design, just like in lipid bilayers, but one side is hydrophobic chain and other side is hydrophilic. And you can make wonderful nanotubes. We know that the amphiphilic molecule first form bilayers, and then two-dimensional material which can scroll to make this kind of nanotube structures. And then the authors, Takuzo Eida, from which group it came from, and so they measured the conductivity. It's very, very high. They call it as a graphitic nanotube, very similar to the carbon nanotubes. So just show this one example to show the powerful nature of this supramolecular chemistry. We can even make materials which can probably compete with the inorganic materials. So there's something, as a prelude I just showed. Let's come back to the supramolecular polymers, which I would like to talk about you. So as I mentioned, if you look into the biological structures, this is again coming back to the microtubule, the video. You can see that this assembly and dynamics, unlike so far the examples I mentioned, where it's a kind of discrete complexes, this is at much higher length scale. Okay, so we can make even nanometer or micrometer long structures if we program the molecules and assemble them to make them. Okay, and so that originated the concept of a supramolecular polymer. Can we have high molecular weight, one-dimensional assemblies just by bringing these monomers together by directional weak interactions? And then probably you can make polymeric-like materials with an added advantage of like reversibility, dynamic nature, and so on. So this was the motivation behind starting the supramolecular polymers. So if you look into just kind of design, it looks very simple, right? So you can make even homopolymers if you have a molecule with complementary groups on both sides. For example, like hydrogen bonding groups like carboxylic acid groups, which is complementary. It can bind itself because it has one hydrogen bonded donor site and a hydrogen bonded acceptor site. So in paper, it looks very simple. So even if you take a phthalic acid, it can form a one-dimensional supramolecular polymer because both sides, it has COH groups, it can form one-dimensional structures. We can also form linear copolymers. If you think about heterocomplementary pairs like this, we can make copolymers and so on. So in paper, it looks very simple, but even like a Jean-Marie Len and others tried in early 90s, but it was not as successful. One of the major problem was these theoretical studies which showed, so you know that the polymeric properties are determined by its molecular weight. So if you want to have an appreciable molecular weight, like in terms of, which is very, very important for its mechanical properties, you should have a reasonably high molecular weight. So let's consider that this is a theoretical study which says that if you want to have 1000 degree of polymerization, that's the number of monomers in a polymeric chain. If you want to have at least 1000 monomers in a polymeric chain, x-axis is the association constant. You need very high association constant. So 10 to the power 7 association constant between the monomers if you want to get a high molecular weight polymer. So this is a big realization, because if you want to use phthalic acid and make supramolecular polymers, you know that phthalic acid association constant, benzoic acid or acid group association constant will be somewhere here 10 to the power 2. So you cannot make polymers. If you start trying to make with acid groups this kind of materials, then most probably you will get a kind of 10 degree of polymerization, so which is not a polymer. So, and if you even take DNA base pair, the association constant is something like 10 to the power 3, and you may not be able to get more than 100 or 150 monomers in a polymer chain. So that was the challenge, initial challenge. We need to identify the self-assembling group or non-covalent interactions which has high strength so that we can make a high degree of polymerization or high molecular weight. So this has been solved by many organic chemists, and they came up with hydrogen bonding groups like this, which is called uridopyrimidones, which has an association constant of 10 to the power 7. And you see that if you have association constant 10 to the power 7, you can get appreciable amount of degree of polymerization in your material. So later people realized that it's not only hydrogen bonding is required, you can also think about host case interactions like cyclodextrin. Host case interactions can be also tuned to make this kind of extended assemblies. So the first part of the supramolecular polymer, although I'm not going to show you the details, was the challenge. The challenge was to identify those non-covalent interactions or those binding motifs which has a very high association constant, so that when you make a material out of it, by attaching these functional groups on both sides of a linear chain or something like that, when you put it into appropriate solvent, it should form a very large polymeric chain. So that was one of the first challenge in this field. So this was well addressed, and people have shown that it has interesting self-healing properties. All of you know this movie. We always realize that self-healing has really powerful. I'm like, if you have a damage in your material, it can come back and form a self-healing materials would be nice. So these supramolecular polymers have shown immense, it has shown enough mechanical properties, some of these uridopyrimidone moieties, which is similar to the classical polymers. And with addition, the dynamics, which I mentioned at the earlier one of the slides, the dynamics helps them to self-repair. So if there is a cut in the material or if there is a damage in your material, the dynamics of the monomers, which goes out of the polymeric chain, will help them to self-repair, self-heal. So these were the initial studies, and people realized that wonderful materials, they can be made completely reversible. So if you apply temperature, you can break them because they are weak interactions. Probably we can use them as a kind of adaptive and self-repairing materials. And then second phase of the system, supramolecular polymers was okay, we can make this big, large number of supramolecular polymers, but one of the challenge was there was no structural control on this material. So if you want to make, so that was the power of polymer chemistry. You can control the dispersity and the length of the polymer to a certain extent in conventional polymers. But when it comes to the supramolecular polymers, it was not easy. Okay, to control its structural properties, this was not an easy job. And so then many groups have started looking into the mechanistic aspects of supramolecular polymerization. We know that polymer chemistry, the mechanistic aspects are well understood. We have two types of mechanism. One is step growth polymerization and the other one is chain growth polymerization. And that which kind of mechanism polymer and by which mechanism the polymers are made plays an important role. Again, we know that because its degree of polymerization and dispersity, the polymer molecular weight and dispersity highly depend on what kind of polymer mechanism you used. For example, if you use a step polymerization, we know that to get a high degree of polymerization, you have to be in a typical condensation reaction, you have to push the reaction to almost like 100% or 99.99% if you want to get a high degree of polymerization. And obviously dispersity control is better here in condensation polymerization. But when it comes to the chain growth polymerization, we know that we don't have a perfect control on the dispersity, but we can get high molecular weight even at any stages of your polymerization reaction. So this is very important, because if you start to making a supramolecular polymer, and if you are not able to get high molecular weight, that means probably the mechanism you should know. Then unless we have a clear understanding of the mechanism, we may not have this structural control. So a lot of effort has been put to understand this mechanistic aspects of the supramolecular polymerization. And the surprise is that similar to the class, as I mentioned at some point of time in my talk, there is a lot of similarities with classical polymerization. So one of the important similarity is in terms of its mechanism. So there were two mechanisms were identified in these kinds of supramolecular polymerization. One is isodesmic mechanism, which is similar to the step growth mechanism. Each step has equal association constant. Okay, so it will be the molecular weight will grow a kind of steady manner. But then if you look into, there is another mechanism which is similar to the chain growth mechanism, where there is an initiation occurs and there is a propagation. Similarly, something which is identified in supramolecular polymers are nucleation growth mechanism or a cooperative mechanism. So there is a energetically unfavorable process of nucleation formation. And then once nucleation happens, the elongation, which is analogous to the propagation in classical polymer chemistry, propagation happens, you get a stack of very high molecular weight. So a lot of attempts has been done to understand what are the important parameters which determine this mechanism. So our group also has devoted almost 5 years of time to understand what are the molecular features which govern these mechanisms. So we had some proposals. Again, I'm not going to the details, but 10 years down the line, we are confident that we can design monomers, program the monomers to follow one of these mechanisms. So that is not a challenge anymore. So the mechanism is well understood. So this is very important, and this insight, the mechanistic insights, help the supramolecular chemists to address next level of challenges in this particular field. So I consider this as an important second phase of, so the first phase was the design of supramolecular polymers, identify the non-covalent interactions, very strong interactions to make these polymers. And the second phase was to understand the mechanistic insights and to identify that in many aspects, it is similar to classical polymers in terms of its mechanism, in terms of its mechanical properties, and so on. So there are a large number of people concentrating on different functions of the supramolecular polymers. Some people are concentrating on the electronic functions. Some of them are on biological aspects. Some of them are to improve the mechanical properties, self-repairing properties. So many aspects, especially to control the rheological properties. So this was the phase, I would say, 1995 to 2005 is a phase where people were trying to synthesize this material. And there was a phase where people talked about mechanisms, like that 5 years was very crucial to understand these mechanisms of polymerization. So with this background, we are now in a position to address the next level of challenges. And when, again, as I mentioned, many times we have to go back to the polymer chemistry and to learn lessons from there. And our group was almost interested in to address the next level of challenges. And one of the challenges to have a perfect structural control. And whenever you talk about a polymeric material, two important parameters is its degree of molecular weight, degree of polymerization. And the other one is how dispersed they are, polydispersity, whether it is close to 1 or it's very high. So up to this point, up to 2010 to 2015, it was impossible to control these structural parameters. So when we started our research group, one of our idea was to understand the molecular features behind these these mechanisms and to control its structural control. Okay. So this is basically, so next probably last few minutes, I'll be trying to tell what are we doing in this particular polymers or in this particular field of research. And this is not only we, I mean, like many groups are trying to address this particular aspect because we feel that it is very important for the subsequent growth of this field, the structural and one more term I have added, which is the temporal control. These two aspects are very, very important for the further development in this field. So when I mean structure, it should have a controlled size, molecular weight, it should have a narrow dispersity. And at some point of time, it's very important that we go from one component to multi-component, right? Two-component system. because in classical polymers, if you look into it, block copolymers form an important aspect in terms of lithography or organic photovoltaics. These are all multi-component block copolymers. So at some point of time, and again, if in nature, there is a sequence control polymerization plays an important role, starting from the protein synthesis with the varying amino acid sequence. So at some point of time, it's very important that we address this sequence as well. So these are the, I would consider as the next level of challenges here. And one more thing which we would like to address is temporal control. Temporal control is an attempt to make more like a biomimetic materials. So what I say is that it's against the same idea of the microtubules. These microtubules are called as dissipative structures. That means they are not equilibrium structures, they are away from the equilibrium. So I'll try to explain what does it mean. So if you look into the most of the monomers, most of the supramolecular materials or any materials for say, they are energetically downhill process, right? They will always try to reach, if it consider an energy landscape, they will try to reach the thermodynamic equilibrium. So during the material synthesis, sometime you get trapped in a high energy state like metastable state, which is true with any material synthesis, but with time, it converts to the thermodynamic equilibrium. For example, the way glass converts to quartz crystals. So if you take into polymers, the supramolecular polymers especially, since as I mentioned, there is a fast dynamics existing in the system. And this dynamics help the polymeric system, supramolecular polymeric system, to reach this thermodynamic equilibrium, right? Because even if it gets trapped in high energy states, the dynamics will help to find the most stable energy minimum. Okay, so it will easily come into the thermodynamic equilibrium. But if you look into most of these filamentous viruses, actin, microtubule, many of the system, they are never under equilibrium. On the other hand, what people say that it is out of equilibrium materials, and you need a continuous supply of energy. You need to pump energy continuously for these materials to exist in their assembled state. In other means, their polymeric state is a non-equilibrium state, and that is very important to perform many biological functions. So as a result, what happens is, so it's an energetically uphill process. The polymers are here, the biomimetic materials are here, they perform function, and then once the energy runs out in the system, they relapse back to the thermodynamic equilibrium. So for example, if you take into microtubule, the GTP binding, which is the energy currency, ATP and GTPs are called the energy currency of the biological systems. So ATP and GTP, when binds into these monomers, they go to this high energy state, and once ATP and GTP get hydrolyzed to ADP or GDP, they relaxes back to the thermodynamic equilibrium. So from a synthetic chemist point of view, it's very important that we move away from thermodynamic equilibrium at some point of time to make out of equilibrium materials. And if we can make out of equilibrium materials, probably that is one step closer towards making lifelike materials, which has a kind of adaptive nature and so on. So this is something, so these are the two aspects, structural aspect and temporal aspects. So once you have such structures, you are creating temporal aspects to the material. That means the material will be stable only for a time, right? Once the energy stored in the system disappears, it will relax back to the ground state. So these are the two aspects we were trying to prove in the case of supramolecular polymers. And if you go back, as I mentioned, many times we have to go back to the classical polymerization. I'm talking about the first aspect, where structural aspect, where we want to control the degree of polymerization, where we want to control the dispersity, and where we want to control the sequence. The kind of polymerization which help to make in covalent polymers this kind of structural control is living polymerization, like ATRP, atom transfer radical polymerization, and ring opening metathesis polymerization. So these are the polymerization techniques which gave a kind of structural control. And nowadays people can make polymers with a narrow dispersity. So can we have living supramolecular polymerization? So in classical polymerization, how it achieved was, because especially no one could imagine free radical polymerization can be controlled. So the way the Machersky and pioneers of these ATRP reactions have done is they have minimized the concentration of the active monomers in the system. So I'm not going to the details. So the way they introduced a kind of dormant state for the monomers in the propagation step. That means you don't have high concentration of the free radical. So that because one of the problem is termination process in radical polymerization. So only way to minimize the termination process is by reducing the concentration of the free radical in the system. And they have achieved by creating a kind of reversible equilibrium there. But then I don't want to go to the details. What was the problem in supramolecular chemistry or supramolecular polymers to have this structural control? If you look into supramolecular polymers, you see that monomers comes and keep growing, right? So there is no termination in principle, right? You can add more monomers. Ideally, it should grow end of it. But one of the problems existed is, which is again outcome of the mechanistic insights in this field. We talk about only now chain growth mechanisms or cooperative mechanisms. So we know that it has two steps. One is nucleation, and then obviously the propagation, nucleation growth mechanism. And although this is energetically, we know that nucleus formation is energetically unfavorable process, but again, most of the synthetic polymeric system, synthetic supramolecular polymeric system, the activation energy for this step is not so high. Okay, so as a result, what happens is most of them undergo spontaneous nucleation. So if you add another batch of monomer into the system, so nucleation happens immediately. So that will kill all structural control in your system, right? So if you add additional monomers, if it starts growing on the existing chain, we can control the dispersity or we can control the molecular weight. But if it starts its own nucleation centers, it will start its own chain formation. You lose that structural control in the system. So one of the ways to do is by having just like again, taking cue from ATRP reactions, their dormant state was introduced at the propagation step. Here, dormant state was introduced at the nucleation step. So what you want is a controlled nucleation. Once you have that controlled nucleation, you can get a structural control in the system. So once you have this structural control, we can come up with a different monomers, and then you can get multi-component structures. So this is the way living supramolecular polymerization has to be addressed. So, but we have come up with a slightly different approach to address this entire system. So I would say this was our major contributions in the last 5 years. We come up with something called fuel-driven assemblies, because we just exactly look into the biological systems, and then we found that there is a dormant system, because if you're taking actin polymerization, there is an actin monomer, which is in a kind of dormant state. It will not grow until it is triggered by the ATP binding. Similar to microtubule, the tubulin monomers will not grow unless triggered by GTP. So it is called a fuel-driven assembly. So this monomer remains in a kind of dormant state. You trigger it at the chemical reaction, then there is a conformational change in the monomeric system. It goes from an inactive state to active state, and it grows. And then there is another chemical reaction which hydrolyzes back this energy like ATP or GTP to ADP and AMP, then bring back the materials to the equilibrium. So we thought, can we have so it's basically a fuel-driven assemblies people have called. So we thought, as a chemist, can we mimic this kind of biomimetic design in synthetic materials, and then probably we'll have structural and temporal control in one shot, because this will be controlling your growth process and this will control your temporal aspects. So we thought, as a chemist, we can have many fuels like enzymatic fuel, reaction-driven fuels, or redox-based fuels. So we try to implement this kind of concept in synthetic materials. So that was the objective. And then I just give you the overview rather than going into each of the system. So one of the things we have done is we have used all the fuels available for us, like enzymatically driven, chemical reaction driven, redox driven. So what we have done is we have created monomers. For example, I will show you one example. So this kind of monomers, this is phosphate receptors. We have taken similar to the actin self-assembly design. So this kind of zinc picoylamine can bind to ATP, or selectively can bind to ATP. So we have taken this kind of biconjugated system and attached with receptors, and then we in situ generated ATP in the system using 2 enzymes. One enzyme from ADP, it can create ATP. The other enzyme, it can hydrolyze ATP to ADP. So exactly kind of actin mimetic system with a multi-component network we have made in the system, where we could really control the growth of this assembly by a fuel-driven assemblies. We can do it with a chemical reaction-driven, redox-driven. Just keep it in mind, it's a kind of fuel-driven synthesis of supramolecular structures. So once we achieve, what we get out of it? So basically, you are controlling the concentration of the fuel, controls the rate of your growth, control the degree of polymerization, control the next key sequence, what you like to come into the system. So you get a kind of biomimetic system. We have that structural control. So as a result, we could make kind of seeded polymerization thereby we can control the degree of polymerization. We can get a dispersity which is close to 1.1 in these structures because fuel controls the controlled growth. And also we can make transient material, out of equilibrium materials. So this is something which we have been continuing in our group. You see that this is a enzymatic network. One is ATP is getting generated, then monomers. So this is a creatine phosphokinase enzyme, which in situ generate ATP in the system. Then these monomers grow into the fibers. And there is another enzyme in the system, which is hexokinase, which hydrolyze back is ATP to ADP. So by carefully modulating the rates, we can populate this particular assembled state, which is an out of equilibrium state. And we can define the lifetime of that state by just changing the concentration of the system. So what I want to say is, by this process, we have a structural control, and then we can make transient assemblies. So you see that just by changing the concentration of this enzyme, we can tell how much time this polymer should be stable. So in this case, it's stable for a few minutes. You change the enzyme concentrations, you can change the lifetime of the material. So the transient materials are those materials where you can pre-program the lifetime. Okay, so these are the, we believe these are the disadvantages one could make by going into a kind of biomimetic designs. So, so this we can address. So what are the advantages of transient materials? We are trying to see that we can think about those materials which are stable only for a time, where the lifetime can be programmed. We can think about various applications like self-erasing inks, transient electronics, where you have, for security purpose, you have a transient circuit. After 1 hour, after conveying the message, it just disappears, just like Mission Impossible movies. There are a lot of possibilities. So this out of equilibrium material is something which we would like to carry on. So before stopping, probably last 5 minutes, I just wanted to say where we go from here. So, so far I show that we can make these supramolecular polymers. To a certain extent, we have the control on the structural aspect. So next few things from a bio-inspired approach, we would like to do it because this out of equilibrium materials can create motion, just like in biological structures. And also the next aspect, similar to the classical polymers, is can we have sequence control in this material? So since considering the time, I'm skipping the motion part of it, movement part of it. Let's come back to the last part, which is the sequence control. So 2 years back, we tried to address this particular aspect because we, as a material chemist, we thought the next functional level, I mean, when next level of functions can be addressed only by going to multi-component system. So we know, as I mentioned, we know that in block copolymers, there's a lot of possibilities in terms of its phase separation, lithography, diblock polymers. It's all well known in classical covalent polymers. And also we know that in inorganic structures, also there are kind of multi-component structures like heterostructures, which is very important for photocatalysis and so on. Can we have axial multi-component system? And one of the special structures we are interested in is kind of axial organic heterojunctions, because we are the people working on organic biconjugated system. If we can create something like that one-dimensional stacks with a P and N type, or different semiconducting molecules, we can get a very nice exciton migration along the stacks, and we can do the same tricks inorganic chemists have been successful in terms of photocatalysis. And we believe that supramolecular polymerization is the only way, a sequence control is the only way to make these kind of structures. So this is something which we call as axial organic heterostructures, or in principle, you look into it, it is a supramolecular block of polymer, right? It has segmented organization of these monomers. So this is something which we have been doing it. And again, we are successful in this particular case. This is a little bit more challenging here because so far I told the dynamics is an advantage, right, in supramolecular polymers. But at the same time, when it goes to one component to two component, this dynamics can be a problem for us. Because if you think about two monomers which we combine together, it can either phase separate like a composite. It can form AB alternating copolymer like a polyethylene terephthalate, or it can also form this kind of supramolecular block copolymer. So the inorganic heterostructure or organic heterostructure we would like to make will be possible if we can control the supramolecular block copolymer. So again, as I mentioned, always we go back to polymer chemistry, and we know that in polymer chemistry, the chain copolymerization is well understood. And that is understood based on the reactivity ratios of the two monomers, right? If the reactivity ratios is infinity, or otherwise AA interactions and BB interactions are stronger than AB interactions, you get always kind of self-sorted structures. And if the AB interactions are stronger than AA, you will get alternating structures, and something in between, which is very close to the reactivity ratio is close to 1 or slightly greater than 1, you can get a block systems or statistical systems. So in terms of, we know that in conventional polymers, the reactivity ratio is determined by the reactivity of the function groups, right? But when it comes to the chain supramolecular polymerization, it's a free energy of interactions between the monomers. So how to do it? So we have also calculated many of the things, and we are working with these kind of molecules. But I just wanted to show one outcome of this. You can see that these are the two supramolecular block copolymers or axial organic heterostructures we made with green and red emissive monomers. These are the monomers. You can see that these are well-known N-type semiconductors. Just by, it's a naphthalene diamide structures. So just by modifying the core, you can get different spectral properties of this core substitute naphthalene diamides. So we use them as monomers, and then we can create these kind of axial organic heterostructures. So we have seen that enhanced exciton migrations. We are collaborating with many physics group to look into the exciton migration in these structures. So what I showed this one example to show that we can even go to next level of complexity. We can now take more than one component and then control their microstructure in such a way that we can make block copolymers. These are unique structures because we couldn't make it before, because we had no control on the mechanistic understanding of the organization. So the last 10 years of increased mechanistic understanding we got in this field, we are now able to address the next level of challenge. But again, I'm talking about only two components. We know that nature does it like a sequence-specific sequence-controlled polymerization in a wonderful way. Amino acid, 20 amino acid, any sequence they can make it. I don't think we have that control yet. I hope in 10 years down the line, we will like to have a kind of sequence-specific supramolecular polymerization. So these were the few. So the last 40, 45 minutes I was trying to tell you is we are trying to go more and more complexity of supramolecular polymers. Just started as a curiosity, probably I would say in 2 decades back. But then it has a lot of advantages in terms of recyclable properties, reversible properties, and so on. So our group is trying to address the structural control and temporal control of these particular systems. With that, I would like to stop. There are a lot of challenges ahead, one of the, especially when it comes to the multi-component system, we face challenges of characterization of structure, because monomer sequence, how do you characterize? That's one of the biggest challenges for us. And whatever the design principle have shown is probably work for a certain class of molecules, it may not work for another class of molecules. So something which we like to have is a kind of predictive synthetic design, just like organic synthesis or polymerization mechanisms. Something which is going on heavily, in a kind of active way in Europe and other places, is using the supramolecular polymers or recyclable plastics. Many times when I give a talk to the polymer chemists, one of the important questions they ask, how do you have a control on the topology? That means, can you make star polymers? Can you make branch polymers? We don't have any control on that. So something which we have started working on it, kind of only way we can have this topological control in this kind of material is by using secondary nucleation, which is again a biological phenomena. We are now trying to make, and also we feel that is very important to compete with the inorganic chemists. If you want to make like a layered heterostructures like graphene, MOS2, and so on, probably we have to rely on this secondary nucleation. So we are working towards that. We have some success, but still too early to talk about it. So with that, I would like to stop by thanking many people, funding sources from many sources, and my students. Collaborations are involved because the complexity of this project requires expert hand in terms of characterization, understanding the molecular interactions by molecular dynamic simulations, functions. So we have many collaborations. I'd like to thank Professor Rao because the unit we are working on, which I joined in 2008, is called a new chemistry unit. And this is a kind of interdisciplinary unit. I'm very happy that this within 10 years, 20 years, this has really emerged as one of the leading departments in the country. I'm very happy to associate with you with it. And thanks to Professor Rao, who had that vision to many people ask me, what is new chemistry unit? It's not a new department, but we are supposed to do new chemistry. So thanks to Professor Rao's vision, and thank you all for your attention, and I would be happy to answer any questions.
No questions. Subi, nobody has posted any of the questions. That's interesting.
Okay.
Anyway, Ivan, you have any question?
Hi, sir.
Hello.
Yeah, like we have many research students, so just for them, may I ask you, like, how do we do
Subi.
characterize part of the two component system?
Hello. So the question is, how do we characterize the two component system, right? Yeah, so this is an important challenge. As I already mentioned, it's not an easy job to characterize as you go to more than one component. So the kind of heterostructures I have shown, I was lucky that to characterize the two component, you see that it has two different emissions. But again, if you go to confocal fluorescence microscopy or anything to characterize, you know that the resolution is an important problem. So in confocal fluorescence microscopy, the 300 nanometer is the resolution. So we have, fortunately, we have a collaboration here, Sarita Gasti, who joined from Rotello group. So he set up something called super resolution fluorescence microscopy. This is coming up to be a very important field. So here we can go up to 40 nanometer resolution. So that's the one which we try to characterize our structures. Obviously, one can go to TEM, because you can use the elemental mapping to characterize these structures. But then you know that unlike inorganic structures, organic structures just burn away. So it was an important challenge for us to characterize this. And then right now, super resolution microscopy is the only way for us. But again, that has a limitation of 40 nanometer or 50 nanometer resolution. So suppose if you have 4 components, 4 monomers in a kind of within 10 nanometer distance or 20 nanometer distance, it's not easy to characterize them. So that's obviously one challenge. That's, as I mentioned towards the end of my talk, the characterization of multi-component system is going to be the biggest challenge for most of us.
Yeah, I think there is another question which is there in the box. That question reads, is aggregation time dependent?
Yeah, so good question. Yes. So the second part of the talk I told about the systems which require structural control, and there we are looking for systems which are kinetically controlled systems, because once it is formed under thermodynamic control, it's nice, they are stable there. So there are 2 types of systems. One is formed under thermodynamic control, one is formed under kinetic control. So the major difference between them is thermodynamically formed system, it forms stable system, but the molecular organization will not change further. Okay, so that's nice for certain applications. But then we are more interested in a kind of kinetically controlled systems, because there comes the control of control nucleation, control growth, and so on. So those systems which I talked about are mostly time-dependent supramolecular polymerization, which are under kinetic control. So it's possible. Yeah.
Okay, there is another question that is from one of our ex-students. She actually is now your neighbor doing research at Christ University. The question goes like this. Hello, sir. Just like peptide assembly on a ribosome, can we engineer supramolecular structures on a similar structure environment like a ribosome or similar pockets?
Yeah. So again, it's a nice question. So obviously, you are talking about again another control we would like to have on a longer run is topological control. So people are working on templated supramolecular polymerization. So it's indeed possible. People have made systems which can be templated and wrapped around the system. It's possible, but we are not working on it. And this kind of topological control, it's the next hierarchical level. Okay, so far we are concentrating on the first level of organization, how to control its structure and sequence. Obviously, as I told, obviously the next question to be addressed is the topological control. Can we make different kind of different kind of topologies? That's something which comes into that, and it's indeed possible. And the only way to do is by secondary nucleation or templated nucleation, which is possible.
Okay. Well, Subi, that was a wonderful talk. You started
Subi,
from the.
Manoj here.
One more question is, I guess, from Manoj.
Yeah.
Yeah.
Manoj, please go ahead.
Subi, one comment, or probably you can take it as a question. For example, at the level of device fabrication, we use techniques like spin coating or dip coating or other techniques. How far we can go towards, for example, your axial organic structures and all. they are quite interesting in terms of photovoltaic devices or other field effect transistors and etc. So how far we can go towards such intelligently designed assemblies?
Yeah.
Imparting it into fabrication techniques.
Yeah. So this is again very good question. So when we deal with the supramolecular polymers, if you want to, I will address them. Like in terms of a photovoltaic devices, it's not a problem. It can be used similar like a spin coating devices or something. That's possible. But when it comes to an FET, what we would like to have is a single fiber transistors, right? Like carbon nanotube or so on. So we need a kind of soft lithography technique. So we have been so even single component system, we have been collaborating with Professor Kulkarni's group to make this single fiber FETs. But then always what we do is, we try to do is like a kind of drop cast and look for single fiber sitting between the electrodes, which is a very crude way of doing it. But then now with this controlled nucleation growth, we are confident that just like carbon nanotube FET is made, we can probably in situ nucleate within the channels, okay, and then kinetically control and bring it, because these are semiconducting system which can be aligned using electric field and in situ growth and control. That's something which we have been trying to do now using this living polymerization to make kind of soft lithography techniques to make fabricates FET, which is an important challenge which we are trying to address in collaboration with other groups. Does that answer?
Thank you, Subi.
Yeah.
Yeah. Thank you, Subi.
Are there more questions from the participants, audience? Seems that there are no more questions. There are no more questions in the chat box. So maybe it's time for us to wind up. Subi, that was a wonderful talk. You started from the first principles and in a way, a wonderful presentation on what you call as bio-inspired chemistry. From very simple things, going for the very complex situation, it's not only like structure. I mean, we understand that everything is a problem. Assembling molecules is a problem. Getting the structure of the assembly is a problem. Assembling this for a particular purpose is a problem. But still, the way you presented it, the way you are doing it, it's certainly convincing that today or tomorrow, you will be coming up with a very useful device or useful methods by which this sort of supramolecular assemblies can be used for excellent biological applications for other technological applications. So we thank you for your kind participation, and definitely we would have liked to have you here. Look for the opportunity to have you here soon, and let's thank you.
Thank you. Thank you very much.
Yeah. I thank you for your wonderful presentation.
Thank you. Thanks a lot. Thanks a lot. Yeah.
Thank you, sir. Before we move on to the next invited talk, there is an important announcement for the participants. The oral presentations are rescheduled to be conducted after the invited talk of Dr. Vinesh Vijayan. I repeat, the oral presentations are rescheduled to be conducted after the invited talk of Dr. Vinesh Vijayan. Hope the participants will cooperate. Now we are moving on to the second invited lecture of Day 3 MATCON 2021. I request Dr. S. Prathapan to chair the session. Over to you, sir.
Thank you. Vinesh? Hello, Vinesh. Vinesh? അടച്ചിട്ടില്ലെന്ന് തോന്നുന്നല്ലോ. Okay, what we will do is, let me invite Litsi. Let her give an oral presentation. Okay, let's see. Let's see, are you online?
Yes, sir. Yes, sir. Hello.
That's okay. If she's willing or she's ready, we can have one presentation for 10 minutes. Let's see. That's
Yes,
a problem. Vinesh, I think, will be joining only at 11:15. So we have a break here.
Yes, sir. Yes, sir.
Since I can take some sort of a liberty with you, since you are directly our student, I thought I will invite you if you are ready, if you are willing.
I'm ready, sir. I'm ready, sir. I'm ready to do the presentation, sir.
Okay. So as you know, The time allotted is 10 minutes, 8 minutes for presentation, 2 to 3 minutes for questions and answers. Positively finish your presentation